Hi everyone, this chapter is going to cover the eukaryote. This is chapter 5 in the Microbiology for Surgical Technologists. So what we're going to be discussing in this chapter is distinguishing between prokaryotes and eukaryotes, discussing the characteristics of the eukaryotic cell, describing the various microbial relationships, and then finishing up with a compare and contrast of the three types of eukaryotic microbes. So a lot of this talk about the eukaryotic cell is going to be review for those of us that just had finished up anatomy. So this will be more like a review for most of you. Remember, the eukaryotes can be one-celled microbes, or they can be multi-celled organisms like humans or the smallest of animals. And the biggest difference between prokaryotes and eukaryotes is eukaryotes have a defined nucleus that's enclosed by a membrane, and eukaryotic cells are a lot more complex. They're also larger. Remember, we said that prokaryotes range from 0.2 to 2.0 micrometers. Eukaryotic cells range in size from 10 micrometers to 100 micrometers. So first we're going to talk about the plasma membrane. Remember, the plasma membrane surrounds all the internal structures of the cell, keeps it together. It's made of this phospholipid bilayer that has these hydrophilic heads and these tails that don't like the water, right? So they hide in the inside, which causes those phosphate heads to attract the water, and they are referred to as semi-permeable. So what this means is one of the jobs of the plasma membrane is to regulate what goes in and out of the cell, and there's a couple ways that it does that. One is by active transport, and the other is by passive transport. Now, when we're talking about passive transport, that means that the cell doesn't have to utilize any of that precious ATP that's been manufactured in the mitochondria due to cellular respiration. Remember when we talked about that? So in this instance, we say that solutes are going to move from a high concentration to a low concentration. Remember, like pushing that rock down a hill. You don't have to keep pushing a rock, right? You just push it and it goes down on its own. But active transport is the opposite, right? It's going to use ATP. That's kind of like pushing the rock up the hill. That's why there's these special protein channels and ion pumps that are housed within the membrane that help to pump those things back and forth. So when we're talking about passive processes, there's a couple processes that we're referring to: simple diffusion, facilitated diffusion, and osmosis. And specifically, osmosis deals with three different types of solutions, and this is the movement of the solute or the solvent, across the membrane. And remember, with cells, there's the majority of the solvent is going to be water. And so we're pretty much talking about water movement here. Some really tiny, tiny particles might slip through, but for the most part, water is what's moving across the membrane. And if we put any type of cell into an isotonic solution, that means there's going to be the same concentration of solutes inside the cell as outside the cell, and nothing's really going to happen as far as water movement. But if we were to put a cell in a hypotonic solution, this means that the concentration of solutes outside the cell is lower, and so that's going to mean that water is going to move inside. And so some antibiotics work this way by damaging that cell wall of the bacteria, and so they can't hold in that water and they explode, they lyse. Right? And then the opposite would be putting some sort of cell in a hypertonic solution, which the opposite thing is going to happen. There's a higher concentration of solute on the outside of the cell, and so water is going to leave the cell, and the cell is going to shrink or crenate, and that's what's going to cause death. And then lastly, what I'd like to say about this plasma membrane is that all plasma membranes of eukaryotic cells are covered with something called glycocalyx, and this is uh, a carbohydrate-based substance that helps the cells to have a stickiness so that they can adhere to one another. So as I mentioned, all eukaryotic cells have a defined nucleus that has a membrane surrounding it and separating it from the rest of the cell. However, different creatures have different cell walls. And here we're talking about algae, fungi, and plant cells specifically. But again, that cell wall is to provide rigidity and shape and protection to the cell. And primarily, these cell walls are composed of cellulose. However, there are some creatures, some cells like mold, their cell walls contain some cellulose, but mainly it's made of a substance called chitin, which is similar to the exoskeletons that we find covering insects, like a hard outer shell. And then only some types of bacteria can break down cellulose. So interestingly, termites, as we know, their favorite food is wood. And termites really can't break the cellulose in wood down either. So they have these little bacteria that live in their digestive system that serve that purpose of breaking down the cellulose so then they can use the nutrients. So they have a symbiotic relationship there. Protozoa, on the other hand, they have these hard calcium shells instead of cell walls, and some protozoa maintain that water balance by having something called a contractile vacuole that fills with the extra water and then they can excrete it. Other protozoa have little alveoli, which we've heard before, right? These little small air sacs instead of cell walls. And then we have our friends, the amoeba, and the amoeba is a tiny single-celled protozoa, and their cell wall is composed of cytoplasm, but the cytoplasm, the outside layer, is called ectoplasm, which really sounds like something from Ghostbusters, and is a clear gel-like substance. And that acts as the cell membrane. And then the inner part is a watery, kind of grainy substance called endoplasm, and that's what contains the organelles. And then one or more nuclei. So let's talk about the nucleus then. So the nucleus is a small spherical structure and it's located in the center portion of the cell. It's the control center. It's where all of that genetic material is located, and it is surrounded by the nuclear envelope. And the nuclear envelope has these little holes in it called nuclear pores. And inside the nucleus is where ribosomes are made. And if you remember, ribosomes are those units that do the transcribing and translating to build those amino acid chains to make proteins. And so they're made in the nucleus, and then they leave and they go out into the cytoplasm, and they help to manufacture those proteins that the cell needs to do whatever it needs to do. And remember when those that genetic material isn't being used, it's really tightly coiled into this crazy mass called chromatin. And then when it is time for the cell to manufacture some more of those proteins, it will unwind itself and the two strands of DNA will separate, and that's where mRNA comes in to the picture and will go in there and make a copy of the DNA, and then it will leave the nucleus through a nuclear pore and go into the cytoplasm where it can be transcribed. The cytoplasm of the eukaryotic cell is also referred to as the endoplasm. And that means that it is the living material inside the cell. And it is surrounded by that cell membrane, and it is this very nutrient-rich substance that houses all of the organelles. And the organelles, that translates to little organs. So inside there we have a whole bunch of little organs, and quickly just to review those, the endoplasmic reticulum. There are two different parts to the ER, the rough and the smooth, and this is where a lot of that protein synthesis is continuing to happen. The proteins are folded and they're folded some more, and some different things like that. And the rough ER is characterized by ribosomes attaching there. And on the smooth, there isn't any room for the ribosomes to attach there and insert their proteins. And so once the protein gets done in the ER, then it is going to move to the Golgi apparatus or the Golgi complex, and that is where the proteins get synthesized even more. They get packaged and they get sent to the membrane of the cell if they are leaving the cell. So we refer to this as the packaging and shipping department of the cell. Within the cell, we also have something called lysosomes, and these are like the trash cans. This is where the garbage goes to get composted and digested by phagocytosis. And then that mitochondria, of course, that's the powerhouse, the power plant of the cell. That's where that very valuable ATP is created via cellular respiration. And then the centrioles lie at either pole of the cell, and their job is to make these chromatin strands. And these are really helpful during mitosis when the chromosomes migrate to the center of the cell, and these strands of chromatin attach to each of those chromatids and pull them to the opposite poles of the cell. And then lastly, the flagella and the cilia are how these cells get around. And the flagella are more like long whippy tails. They're more complex in the eukaryotes than the prokaryotes. And then there's the cilia, which are the little tiny hair-like projections, and they're not just on microbes. We also have them in our bodies as well, lining our respiratory tract that help to move those foreign particles up and out of the respiratory tract. So inclusion bodies are really commonly found in prokaryotic cells and help to store extra nutrients and energy and those kinds of fun things, but in eukaryotic cells they are very uncommon and often point towards some sort of abnormality, such as a genetic abnormality or some sort of mutation or viral invasion, something like that. And so some examples that the textbook gives are Russell bodies, which are associated with cancer, Negri bodies, which are associated with rabies, Garnier bodies, which are associated with smallpox, and Cowdrey bodies, which are most closely associated with herpes and the polio virus. Now, there's different types of microbial relationships, and when one organism lives in close proximity to another, we call this relationship symbiotic. And there are three different types of symbiotic relationships. The first one is mutualism, and mutualism means both organisms are benefiting from the relationship. A commensalistic relationship is where neither are benefiting or being harmed, or one is benefiting while the other is not being harmed. And then lastly, a parasitic relationship where one is benefiting, the parasite, and the other is receiving harm, which would be the host typically. Now, there is a theory associated with symbiosis, and that's called endosymbiotic theory, and this means that scientists believe about 1.5 billion years ago that these organisms that were living in really close proximity to one another one just happened to engulf the other, and they had this mutualistic relationship. And so instead of that organism that was engulfed being destroyed, it went on to become a part of the functioning organism. And similarly, the first eukaryotic photosynthesizers, they think they came to be in a similar fashion. Now, there are some different types of protozoa, and these protozoa, if we're referring to one, is a protozoan. They typically live in watery environments. Now, if there's a situation where the environment becomes uninhabitable, they can turn into a protective little version of itself called a cyst, similar to how some bacteria can develop spores. They are referred to as heterotrophs, and they have this special little organelle inside themselves called a vacuole that serves to digest their nutrients. It's like their stomach. And they move via three different ways: flagella and cilia, we talked about before, but there's another way called a pseudopod, and that means a false foot. And so some examples of protozoa that do move about are flagellates, amoebas, ciliates, and sporozoans. Okay, so let's talk about algae and fungi. So algae, like protozoa, they also like a wet environment, and there are many different types of algae. Um, there are they are photosynthesizing creatures, and they are considered as autotrophs, and they contain something called chloroplasts, and the chloroplasts are what determine what color they are. And red and green and brown are typical colors of these chloroplasts. And the red-brown types are typically marine species. And at first they thought that cyanobacteria were a type of algae, but they realized that they're really just prokaryotic microbes, and they're the blue-green kind. And so you might have heard of something called plankton, and plankton is there's two different types of plankton, and this is what is food in the ocean for a lot of animals that live there. And two types are called diatoms and dinoflagellates, and these are responsible for causing something called algal blooms, commonly happening in lakes and streams. And sometimes, if you've ever referred to something as pond scum, this is what you're seeing are these planktons. And sometimes they can become harmful. If they do kind of take over the place, then they can deprive the life living there of oxygen, and they do release some toxins that can also be harmful. Now, while they're not pathogenic to humans, the toxins that they produce can get ingested by the fish and the shellfish that live there, and then if we eat that, then we can get sick from that. And then lastly, fungi. You know, we can't talk about eukaryotes without talking about the fungi. And when I hear the word fungi, I think mushroom, right? Shrooms. And yes, that is the Latin word for mushroom, but a lot of them are microbial forms such as molds and mildews and yeasts. And scientists believe that there's probably 1 million species here on Earth, but they've only documented 100,000 so far, 400 of which are pathogenic to humans, but far more, 5,000, that cause diseases in plants. They are considered heterotrophs. However, they cannot perform photosynthesis, and therefore that's why they like living in these dark, moist areas. And fungi can be of help to us. And you're probably familiar that with yeast and its uses in making bread and different types of alcohol. And then mold played a big role in making our first antibiotic, which was penicillin. Okay, so let's just review here. So let's look and see what we have in this top image here. What do you think that is? If you said protozoa, you would be correct. Remember, these little protozoa love water, but in harsh times, they can turn into a cyst until conditions get better. Top middle, we're looking at some of those algae blooms, beautiful green algae blooms. And then this last one on the top, what do you think that is? If you said fungi, you would be absolutely right. Now going to our middle left image, what type of relationship do you think we have there? If you said mutualistic, you would be absolutely right. The hummingbird benefits by getting the nectar from the flower, and the flower benefits because the hummingbird gets some of that pollen on its beak, and then when it goes to the next flower, it helps with pollination. That middle image there, what do you think that is? If you guessed Golgi apparatus or Golgi complex, you would be absolutely correct. Remember, this is where the proteins go after they leave the ER. They come to the Golgi apparatus to be packaged and then, if necessary, sent to the outer membrane or sent to the membrane of the cell to be shipped outside of the cell and to wherever its destination is. What about this middle one on the right? What type of relationship do you think? If you guessed parasitism, you are absolutely correct. This is a tick and his host, his or her host, I don't know. And clearly the tick benefits where the host is harmed. And then this bottom left one here, this represents commensalism, right? The birds are benefiting, the horse isn't harmed, he gets rid of some pests, right? But doesn't really care. Bottom middle, what do you think about that one? If you guessed rough ER, you would be absolutely right. Remember, the ER is where there's more, that synthesis of proteins is carried out, and the rough ER is rough because those little purple dots are representing the ribosomes, and they can attach there and insert their proteins. And then lastly, our bottom right is resembling phagocytosis. And remember, this is the engulfing of some sort of foreign invader and by the process of phagocytosis so that that can get digested and excreted from the cell. All right, well, I kept it under 30 minutes. This is the end of chapter 5. I hope you thought it was helpful. I hope you found it a little bit interesting, and I hope you're one step closer to falling in love with microbiology.